So this is lesson 2. Lesson 1 is the sheet you did yesterday with the scientists and what they found on it. Because this is Friday and I want to do this, we might go over that sheet today, and if not, we will go over it in our class next week. So we're talking about DNA and RNA. The first thing that I would like to discuss is what DNA is actually made out of. So you'll notice on the sheet that you have a picture of DNA, and then you have this exact picture, which are nucleotides. Nucleotides are made out of a few things. Each nucleotide, so there are 4 different ones, they each have some pieces in common. For example, they are made out of a 5-carbon sugar. So if I look at the structure, everywhere that you see a D, it looks kind of like a pentagon. That's your 5-carbon sugar. Each of the corners is a carbon. And the name of the 5-carbon sugar is deoxyribose. So that's why there's a D. Now normally I would say, oh man, the detailed name of chemical molecules is kind of beyond the scope, but this is a spot where I think it's important. And the reason it's important is because when I show you RNA, the other version, this part, the 5-carbon sugar, is what is different. And so I'm highlighting this part here. DNA nucleotides also have a phosphate group. And so the phosphate group, anywhere you see a P, that's your phosphate group. Phosphate and phosphorus are not the same thing. Phosphorus is an atom. Phosphate is phosphorus and oxygen together. That's beyond what you would be expected to know, but I'm just telling you, in case you're in chem, phosphate and phosphorus, PO4, PO4 3 negative. If you were wondering. Does anyone else want to quiz me on my periodic table knowledge? The other part, and this is the part that distinguishes each nucleotide from each other, the other part is the nitrogenous base. So you'll notice on the top part of each of the nucleotides, there's a structure. Sometimes it has 2 pieces, sometimes it has 1, and I'll talk about that as well. But there's a structure that has an A, a T, a C, or a G. So the A stands for adenine, the D for thymine, the C for cytosine, and the G for guanine. A, T, C, G. Now you might notice perhaps that A and G have something in common. When I look at the top of their structure, they have sort of 2 shapes, a pentagon and a hexagon, whereas T and C look similar in the sense that they only have 1 piece. We call T and C, they are both part of the same category, and they are called pyrimidines. And you can recognize they look similar, right? They have this, the top part of their structure looks similar. The other 2, A and G, so adenine and guanine, look similar in the sense that they have the 2 pieces on the top, and they are called purines. So of my DNA nucleotides, I could split them into 2 groups, pyrimidines and purines. And one of the things that we'll see is that when DNA actually pieces together, pyrimidines and purines like each other. They like to pair up. So, you know, opposites attract. 2 purines wouldn't pair up with each other. And if you look, it makes sense size-wise. If 2 purines paired up, there would be a bigger space. If 2 pyrimidines paired up, the space would be smaller, and then your DNA molecule would be changing size. So when DNA pairs up, A always pairs up with T, C always pairs up with G. So you might ask yourself, how in the world am I supposed to remember some ridiculous thing like this? So I'll tell you how I remember. When I was in grade 12, in my class, there were two people named Adam and Tara who were dating each other. And I was like, oh, sweet, Adam and Tara goes together. And by miraculous chance, their last names were Cracknell and Garvin. So it worked out magically perfectly. You might think of some other funny little trick to remember that A goes with T and C goes with G. What did you say there? Adam and Thomas? So like his first name and your last name? Oh, that's sweet. That's sweet. That's really sweet. Oh, he meant this Thomas. I thought he was talking about his last name. Or any other thing that you would like to remember them by. You'll notice as we get through this unit, you're probably not going to forget that A goes with T and C goes with G, but you might want a little memory trick just so when you get to the test and you're like, oh shoot, does A go with G or with C or with T? And if you have that little memory trick, it'll jog you right away and you'll feel confident that you're not forgetting. Does anyone want to ask anything here so far? So what this means then is that A and T have to have the same proportion. So however much A there is, however much adenine there has to be the same proportion of thymine, and the same would go for cytosine and guanine. They have to be present in the same proportion because they're always going to pair up with each other. Now the two sides of each pair we would refer to as complementary bases. They complement each other. One has two rings, the other only has one, and when we put them together it makes a beautiful three-ring structure. And the same goes for A and T. So C and G complement each other, A and T complement each other. Now, does anyone have any questions they want to ask so far about DNA nucleotides? So these are the tiny little individual units. We know that DNA can go together in a big long strand. So what would that be like? The DNA nucleotides that we just saw go together in a strand, in fact, two strands to form a double helix. So double means two. Helix is the idea, oh, that might be the nicest helix I've ever drawn. Helix is giving you the idea of the twisted shape, and double means two. The double helix then is two strands that are twisted over each other. If you think of where you might have seen heli or helix, that beginning part of the word, like in a helicopter where the blades spin around. So double helix is talking about the twisted structure. If you take a look at your picture or what's on my screen, you'll notice then that there are two strands that they cross over each other right there to make the twist in the helix. And you'll notice that if you take a look at the legend, there are phosphates and there's deoxyribose. You'll notice here there's a D and a P. So the deoxyribose is kind of the thicker part and the phosphate is the thinner part. We would refer to this as the backbone. So the backbone is sugar and phosphate. So along the edges, you would see alternating sugar and phosphate because each DNA nucleotide has a sugar and a phosphate. So every time I add a new nucleotide, I add a sugar and a phosphate. Yeah, so creatine phosphate has phosphate in it. The other part of the molecule, the creatine part, isn't the same as deoxyribose, but the phosphate certainly would be. So along the edges of your DNA, you have the backbone that's made out of alternating sugar and phosphate. On the inside, what I would like to call the rungs, and this is not like an official scientific vocabulary, I'm comparing this to a ladder, a twisted ladder. So if we look at DNA, what it really looks like is a ladder that you've twisted up to make sort of like a circular ladder out of. The rungs are actually complementary bases. And you might notice they are not concretely attached to each other. You see those little dots that are in between them? The complementary bases are attached by hydrogen bonds. Now, if you didn't take Chem 20, then you might not know what this is. So I'm just going to give you a brief description. A hydrogen bond is an intermolecular force. It's attraction between two molecules. It happens when, essentially, hydrogen is bonded to something that is very electronegative and takes the electrons very close to it, so hydrogen is left with essentially none. It gives hydrogen an almost full positive charge, even though it's part of a molecular compound. If you just listen to me and were like, what? I hate chem, that's why I took bio, then just recognize we're not going to ask you to explain in detail what a hydrogen bond is, but I just want you to see it's an attraction. It's not like they're physically attached to each other, but they're attracted to each other because of this hydrogen bond. Hydrogen bonds are what holds, for example, molecules of water together so that they flow, keeps ice as ice. And if you look, they're always complementary. So adenine is always paired with thymine. Cytosine is always paired with guanine. Does anyone have any questions that they want to ask so far? Yeah. So the question is, could incorrect bases, I'll just move this out of the way, could incorrect bases be attached to each other? And the answer is not really, because the hydrogen bond won't form properly in between them. It's possible for a person's DNA, let's say, to have a mistake, to have a mutation. If it's just one nucleotide, sometimes it doesn't change a lot about the structure of DNA, but it will certainly change what we do with the DNA. And so when we get to the end of this and we talk about mutations, you'll see what the effect of changing one nucleotide would be. But essentially, they have to be paired up this way. It won't be attracted properly if it's the wrong nucleotide. Yes, Morgan. Yes, so in chem, to make it a hydrogen bond, the hydrogen would have to have been bonded to an O or an N or an F, because those are the really electronegative atoms. So these are nitrogenous bases. So there's actually nitrogen and oxygen in all of them. Does anyone else have any questions they want to ask? The other word that I want to mention, and I'll write down here, is that the two strands are anti-parallel. And so I'll just explain sort of what that means. If I were to look at, say, the top and the bottom strand, they go in the opposite order. If one starts with phosphate, then goes sugar, the other starts with sugar and then goes phosphate. So when we talk about anti-parallel, we're really referring to the sugar and the phosphate in the backbone and how they're organized. Now the picture that's on your coloring sheet is sort of a cartoon version of it. I put another picture here to help explain what anti-parallel would look like. If you take a look here, like on the last picture, the sugar is like the pentagon looking part and the phosphate's the little circle sticking off the side. And if you look, the strand on the left has the phosphate and then the sugar and then the phosphate and the sugar, and the other one looks like it's turned the other way. So like literally the orientation is upside down, and you can really see it with the sugar. You can see that the point of the pentagon is pointing down instead of up. Yes? It's just how many hydrogen bonds are actually forming between them. Not really, no. It's just the way that the molecule is oriented, how many attractions form between the two things. Does anybody else want to ask anything here about double helix, backbone, anti-parallel, the fact that it's complementary? No, not really, no. Okay. So the other type of molecule that we will discuss in this chapter is RNA. Now I put next to it mRNA because there's actually different types of RNA. The little m stands for messenger. And I put that on this slide because that's the one that the picture is actually showing. We will talk about Well, we'll talk about 3 in detail and a 4th one that I'll mention, but there are lots of different types of RNA, and it's basically dependent on their function. What are they doing? They're made of the same thing, though, and this picture, which represents mRNA, illustrates the most clearly what they're made out of. mRNA looks really similar to DNA in that there is a sugar phosphate backbone, and that there are 4 nitrogenous bases. So far you would be like, well, what in the world is different between this and the last one? So the first very obvious visible difference is how many strands. There's only one. So DNA is a double helix, mRNA only has 1 strand. And that would be true for all the RNAs. They are single strands instead of double strands. The second thing that's different is that the sugar is not deoxyribose, but rather ribose. So you'll see on the picture, there's a P for the phosphate, but then there's an R. That's the sugar, that's ribose. So it's still a sugar, but it's just a slightly different version of the sugar. The other thing that's different is that one of the nitrogenous bases is not the same. So instead of thymine, there is uracil. It pairs up with adenine, just like thymine would, but it's different. So we don't have thymine, we have uracil. So you'll notice when you color the nucleotides, one of them is different than it was on the DNA. This is very often the thing that a question might focus on when asking you, is this DNA or is this RNA? What does, sorry? This P right here? What's the pH of RNA? Well, it's based on nucleic acids, so I don't actually know offhand if I measured it, what it would be. I think it would depend on which cell it was in, but it's probably somewhere close to neutral because most things in your body, with the exception of things that act in your stomach, function best in neutral. Probably a little bit higher than 7, that's what your blood is. Anybody else have questions? I'm going to have to look that up today now. So that's your RNA. It looks like DNA except that it has those 3 key differences. So I said there were types of RNA, and I just want to mention them now, and then we'll talk about a couple of them in a lot more detail, but I just want to give you a brief overview of what the types of RNA are. The first one is the messenger RNA. Messenger RNA, like its name suggests, takes information from the nucleus, usually to a ribosome. And then you're going to say, oh shoot, what is a ribosome? I know I've heard that word before. Does somebody remember what a ribosome is? Do you remember or do you have a question? Yeah. Oh, there you go. I was wrong. 4 and 5. Yeah. Yeah, a ribosome is the structure that helps accomplish protein synthesis. So if you remember learning or hearing about ribosomes, this is a cell. It's awesome. In the cell, you have a nucleus, and around the nucleus is the endoplasmic reticulum, which I've drawn exceptionally well. And on some of the endoplasmic reticulum, there were little dots. Those are ribosomes. Ribosomes can also exist just out in the cytoplasm, but ribosomes is a structure that's part of a cell that we will talk about quite a bit because messenger mRNA is going to take information from your nucleus of your cells to the ribosome. When we get there, we have ribosomal RNA, so it sort of makes up the ribosome. You'll see that a ribosome isn't really a dot. It's actually a sort of two-piece structure. There's a piece on the top and a piece on the bottom. And the messenger RNA will get fed through the middle, kind of like we were reading the information off of something. So if you've ever used one of those machines, where you put what am I trying to say? Paper money, a $5 or a $10 bill in a machine, and then it gives you change. What it's doing is it takes that piece of paper money, figures out what it is, and then gives you the money as a result. We could compare what's happening to messenger RNA with that. Transfer RNA brings amino acids to the ribosome. Anybody remember what an amino acid is? Yeah? Yeah, it's what you would get if you broke down a protein. So the point of basically your DNA and all of your RNA is to code for a protein. Your DNA is in your nucleus and has some information. Your messenger RNA takes that to the ribosome where it gets read. Transfer RNA brings amino acids, and in the end, we end up making a protein. Now, this is the very brief overview. We will talk about all of the details of this process and how that works, but I think I have two more words that I want to talk about, and that is the end of today. You will hear or you will read about DNA sequencing. This is referring to the order of nucleotides. Does it go ATCG or does it go AAA, TT, GC, GC? That is what DNA sequencing is. A gene, we already saw, but I'll remind you, is a section of DNA, we knew that, and I'll add that codes for a protein. That is the point of this whole process. Your DNA codes for proteins. The proteins go out and do things in your cells. A genome is the entire collection of nucleotides in an organism. So a gene is small, genome is quite a bit bigger. You might have heard reference to things like the Human Genome Project, trying to map every chromosome that humans have. I don't know how that happened. Trying to map every chromosome that humans have to figure out what all of the gene possibilities are.